It's my pleasure to introduce Dr. Frank Delaglio. Dr. Delaglio is a principal investigator of the Biomolecular Structure and Function Group at the National Institute of Standards and Technology. He's a leader in developing computational methods to analyze and extract useful information from NMR data. In fact, he's the developer of NMR pipe, which is routinely used for processing NMR spectra. Our lab has collaborating with him for the last few years. This year, our lab published a paper with him on the time domain-based spectral modeling method, which is SAND. And folks in our lab use SAND on a regular basis. My research, in fact, is to apply SAND for peak modeling and quantification in time series NMR. Students like me who know him personally can tell what a great experience it is to learn NMR signal processing from him. And he's so good at explaining complex NMR concepts to the audience. And so without further ado, please help me welcome Dr. Frank Delaglio.
Thank you for that splendid introduction. All right, very good. So, uh, today I'm going to give an overview of the kinds of work that our NMR groups here are involved in. And I'm speaking to you from a little institute called the Institute for Bioscience and Biotechnology Research, which is in a Maryland suburb of Washington, D.C. And the Institute is interesting because it's jointly run by the federal government, that is to say, the National Institutes of Standards and Technology, and the University of Maryland. It's on a small campus of the University of Maryland. There are about 200 investigators here. And even though the Institute is very small, we are well endowed with biophysical measurement techniques. We have 900 megahertz NMR, cryo-EM, X-ray crystallography, a state-of-the-art small angle X-ray scattering system, and experts who do new methods development in mass spec for proteins. So all of those different areas are things that we do in the Institute, and we generally have postdoctoral opportunities in all of these different areas. Today, I'm going to talk about the work of the NMR investigators in the Institute, and in particular, work that's aimed at supporting biomanufacturing. In a general sense, the things at the Institute focus either on general structural biology, which you're all quite familiar with, or biomanufacturing. And by biomanufacturing, I mean the production of protein therapeutics for live cells and the production of live cell therapeutics such as CAR T. Now, NIST is very interested in developing measurement methods and standards. And so, as you'll see, this is a strong emphasis in the work that we do. Just as a little bit of a reminder, much of the motivation for the work that we do, both in structural biology and biomanufacturing, is to support the development and manufacture of drugs. And we can think of drugs in two very broad categories, being small molecules and everything else. Now, small molecule drugs, like aspirin, for example, everything there is to know about them is defined by the covalent structure of the molecule. And when drug companies try to find a new small molecule drug, generally they have a protein target associated with a particular disease state, like here, cyclooxygenase, and they try to find molecules that will interfere with the functioning of that protein. So, for example, this particular enzyme catalyzes the production of prostaglandins, which promote inflammation, and also thromboxanes, which promote blood clotting. So if you can interfere with this enzyme, you can improve both inflammation and reduce blood clotting. And generally speaking, small molecule drug design will start with a small molecule that shows some activity, and then put that small molecule in a building with 1,000 medicinal chemists and have them make different derivatives, add different molecular shrubbery to a basic scaffold and try to find a better drug. And to a large extent, a lot of this is trial and error. What you see here are a collection of different molecules that can all interact with this particular COX enzyme. The challenge is that if there are so many different molecules that can interact with the same enzyme, it also means that chances are for a given small molecule drug, somewhere there's another enzyme that's going to be affected that we don't want to be affected. So these off-target effects are often why candidate drugs fail. That said, to some extent, finding small molecule drugs is a numbers game. It's based on how many small molecules you can actually try against a given target. And a large pharmaceutical company will have several million small molecules to draw on. Now, on the other hand, the fastest growing category of therapeutics is monoclonal antibodies in the class of protein therapeutics. And these are unique compared to small molecules in several ways. But as we mentioned in the small molecule case, it's a numbers game. We want to have a large molecular diversity to try to find a small molecule that can hit a particular target. In the case of antibodies, which have these Fab domains that can bind antigens, a given individual generates 10 billion different structures of antibodies. And so a much huger source of molecular diversity than any drug company can bring to bear with small molecules. And for this reason, monoclonal antibodies are very attractive drug platforms, not only because we can generally find a particular monoclonal antibody that binds well and specifically to most any target, but also all antibodies within a class are relatively similar to each other. And so the details of how to express them, purify them, test them, and deliver them are largely similar. And information that we learn about one particular therapeutic antibody can often be extended to improve our understanding and capabilities with other antibodies as well. So for this reason, monoclonal antibodies are the fastest growing drug platform. They account for 5 of the top 10 drugs, sales of over $150 billion a year. So these are incredible medications. They are life-saving and life-changing medications. Recognizing this point, NIST, which loves to make reference standards and data, in partnership with a pharmaceutical company, created the NIST-MAB, a reference monoclonal antibody. And it's been characterized by every possible biophysical technique that you can think of. So now this NIST-MAB reference antibody is a platform for methods development for manufacturing of MABs. We're going to use two-dimensional NMR as a way to probe the structure of a MAB therapeutic. Now, as we mentioned for small molecule drugs, when you know the covalent structure, you know everything there is about the molecule. But in the case of proteins, proteins have a particular amino acid sequence, but that sequence has to fold into a particular shape in order for the drug to have its proper activity. And if the drug is misfolded, it might ruin the drug's activity, or potentially even more seriously, the drug might maintain its activity, but cause immune responses because it presents a different shape to the immune system. So a critical need when we manufacture these protein-based drugs is to ensure that the fold of the protein is maintained. And in pharmaceutical circles, they call this overall structure the higher order structure of the monoclonal antibody. And we're going to use a very straightforward idea that will measure a spectrum of two molecules. And if the spectrum is the same, the structure of the two molecules is the same. That is the straightforward idea that we're going to apply. And for the most part, we're going to use a spectrum like the one you see here, which is a 2D proton carbon spectrum. And it's displayed here as a surface, but more often we display it as a contour plot like this, where each one of these little blobs is a peak. Each one of these peaks is associated with a CH group somewhere in the molecule. The position of that peak in the horizontal dimension is the NMR frequency of a hydrogen atom, and the position of that peak in the vertical axis is the NMR frequency or chemical shift of the associated carbon atom. So if the shape of this molecule changes, these peaks will move around. They'll get bigger, they'll get smaller, they'll get narrower, they'll get broader, they're split, they'll disappear. So characteristic changes in the spectrum associated with characteristic changes in the structure of the molecule. Generally speaking, when we're working with a therapeutic, we don't have isotopically labeled materials in the same way that we do when we do structural biology by NMR. So usually we don't know the specific atomic assignments of peaks in a spectrum like this, but we do know broadly what kinds of amino acids cause the different peaks. And as needed, we can find specific assignments, but we often don't have them. So what can we do when we have 2D spectra like this, but we don't have assignments? How can we monitor higher order structure? Well, the reproducibility of making these measurements was studied in a multi-lab study arranged by Rob Brinson here. And in this study, we sent the same two MAB samples to 26 different labs around the world with many different kinds of spectrometers and had them reproduce the same measurements. And what we found is no matter who does the measurements on what kind of spectrometer, the chemical shifts of the peaks in these spectra are reproducible to better than 6 parts per billion. So the positions of these peaks are exquisitely reproducible. And so if we have an analyst choose, say, about 100 different reference peaks and tabulate their locations, we can do all sorts of analysis that reveals details about the underlying structure and how it was measured. The challenge is, in this study, there was a collection of 375 spectra produced, and there are about 100 reference peaks. It takes an expert a very long time to be able to analyze a collection of data like this. And the analysis is somewhat subjective because the analyst has to make decisions about what peaks are what. And an analysis like this one, where we're looking for a particular set of reference peaks, is not useful for comparing two spectra that are very different from each other and have potentially very different numbers of peaks. So this analysis method of going on a peak by peak basis is very powerful, but it's also subjective and really time consuming. So we'd like to find other ways to be able to extract information like this without the need for extensive interactive work by an expert. And as a typical example, what you see here on the left are spectra measured on two different kinds of samples. One spectrum is in red, the other spectrum is in blue. And as you can see, they're almost identical to each other. And a similar case on the right-hand side, two different samples, one in red, one in blue. Can you look at these spectrum and tell whether or not the spectra are the same or different? It's not easy even for an expert to do this. So what we would like to do is come up with mathematical procedures that can directly reduce a collection of spectra measured on two different samples to an analysis result like this, where all of these objects in blue represent replicate measurements on one sample, and all of these points in red represent replicate measurements on another sample. And these are uncertainty bounds. So we can look at a graphic like this and tell very clearly these two samples are substantially different from each other according to their spectra, whereas these two samples are not. So very useful. How can we make an analysis like this one? We can do it with a method called principal component analysis, or PCA. And to demonstrate how this method works, we're going to look at a particular case of a forced degradation of the mAb, something that pharma companies do all the time to test how well a particular antibody therapeutic will survive on the shelf and retain its stability. One of the things that can happen, one of the post-translational modifications that can happen, is oxidation of methionine residues on the surface of the antibody, which changes how the antibody can interact with the antigen and also changes how the antibody can interact with the receptor on the immune cell. So in this example, we have two kinds of mAbs. In one case, we oxidize it with peroxide directly. And in another case, first we bind the antibody to protein A that binds to a specific location on the antibody surface. And it just so happens that underneath that location are some methionine residues. So some of the methionine residues will be protected from oxidation when the antibody is bound to protein A. We have these two kinds of samples now. Now, if we look at the spectra, in this particular case, the two samples that we made are almost completely oxidized. So the methionine peaks are completely gone. We have to look very low in the contour levels. So in blue is the only signal that's left over from the antibody oxidized all by itself. And in red are the tiny little signals left over from the antibody oxidized in the presence of protein A. And since protein A protects some of the methionine somewhat, we have a few more signals in this sample than we do in the other, exactly what we expect. Now, when we use principal component analysis, which I'll explain in a bit, we get two kinds of results that we can inspect and exploit. One result is a scatter plot that we've seen before, where each item in this scatter plot represents one spectrum in a spectral series. And another thing that we can get from principal component analysis are synthetic spectra that are associated with each of the dimensions of these scatter plots. And as I will show you, a spectrum associated with the first principal component is very much like an average spectrum, and the spectra associated with the remaining components are going to highlight the parts of the spectrum that are different, that vary between these different kinds of samples. So first component here, which is drawn in gray, you could think of all the places where the samples have intensity in common. And the parts that are drawn in red and blue are parts of the higher components. And those are parts of the spectra that are different between these different classes. So how does this all work? Let's look at a little simple simulated case where we have two replicate sets, just like we're talking about. The spectra have one peak in common and one peak that's a little different. and we have a bunch of replicates of each one. So we have a spectral series like this. In principal component analysis, we take the spectral series being analyzed and we make a linear combination of the spectral series. So we find a set of coefficients such that when we multiply them by each of these spectra and add them together, it generates a single 2D spectrum. And we adjust these coefficients such that the 2D spectrum that we generate is the best match in a least square sense for all of the spectra in the series. So we make a linear combination of all of these in a way to make the best approximation for the entire series. And that's the first principal component. The coefficients are the score values that we use in the scatter plot, and the linear combination that we generate from these coefficients is the loading spectrum. Once we have this loading spectrum, we can subtract it from all of the original spectra, and that generates a residual, and we can repeat the same procedure and find a new set of coefficients such that when we use these coefficients to make a linear combination of all the residuals in this series, we generate a new component spectrum that best matches this series in a least square sense. So when we have a series consisting of two kinds of samples, we expect that two components should be sufficient to describe the entire spectral series. And generally speaking, this first component looks very much like an average spectrum, and the second component looks very much like a difference spectrum. If there is no change to the underlying materials, two components should be enough to describe two kinds of samples. But if the measurement drifts or the samples change during measurement, then we will have signals in the higher components. So seeing signals left over in the higher components for a two-sample system is diagnostic of some change during the course of the measurement. Now, here's an interesting aspect of principal component analysis. To generate a two-dimensional data set, we measure a bunch of time domain data, one-dimensional data sets. We Fourier transform all the rows of these things, and that generates an interferogram. And then we Fourier transform all the columns of this, and that generates our 2D spectrum. In NMR, we have a method for reducing measurement time by skipping over some fraction of these 1D measurements that we make. What that means, though, is that when we try to Fourier transform this kind of data that has gaps in it, places where we skipped over a measurement, the spectrum that we get has unusual artifacts, and we can't easily do it directly. But the Fourier transform doesn't change the information content of the data. It just rearranges it in a linear transform. So from the point of view of principal component analysis, the information in all three of these cases, time domain, interferogram, and 2D spectrum, are all the same. What that means is we can apply principal component analysis to a spectrum, but we can also apply it to an interferogram or some other form of data that can't be visually analyzed, like this case, which is the result of a non-uniform sampling measurement that's been Fourier transformed by conventional processing instead of the special reconstructions that we usually need. So we can get the same kind of information from all these data sets. So principal component analysis gives us a way to extract information from spectra where there might not be features that we can identify as peaks. And that's rather powerful. And now to show you an example of what we can do in the context of actually understanding the behavior of a therapeutic and characterizing it, as we mentioned, it's important to study the oxidation of these methionine surface residues because that changes the way the antibody interacts with its targets. So, for example, if we look at thermal unfolding times with increasing amounts of oxidation, these thermal unfolding times get smaller and smaller with longer oxidation, meaning that the protein is becoming less and less stabilized as more and more oxidation occurs. Furthermore, if we monitor the binding of different kinds of proteins like protein A or protein F, these two proteins bind in positions that protect some methionines on the surface. And so, since the methionines are in the binding site, increased oxidation time reduces the binding affinity with these two peptides. Protein L binds to a place where there's no methionines, so increasing oxidation does not change its binding affinity. Furthermore, another thing that is pioneered by folks here at IBBR is the use of relaxometry, water NMR, to characterize biopharmaceuticals and vaccines. And this is work pioneered by Bruce Yu and Mark Taraban. And as you see in this example here, these are water relaxation measurements on these samples with respect to oxidation time. So you can see the increase in relaxation rate that's associated with destabilization of the protein. So all of these biophysical methods that can be used to gauge the activity and stability of the underlying mAb. And now look at what happens when we correlate them with the principal component analysis scores. So in all of these cases, the relaxometry data, the activity by SPR, the stability by thermal unfolding, all of these correlate directly with the principal component analysis score values. So we can get quantitative information about activity and stability from the NMR data, and it didn't require any peak analysis. So that's pretty cool. And that's an example of the kind of work that we're doing to support characterization of therapeutic high order structure by NMR. The NIST mAb became NIST's most popular reference material because of the importance of monoclonal antibody therapeutics. And of course, these antibody therapeutics are generated by bioreactors with live mammalian cells. So, as a follow on reference material, NIST has produced this unique live cell reference called the NIST CHO, which is a CHO cell that's been engineered to express the NIST mAb. And it's been engineered in the same way that a pharmaceutical company would engineer a cell line to maximize the amount of protein that it can produce. So this reference cell line was made specifically to mirror a realistic case for pharmaceutical manufacturing. And so now we have a test bed for what happens in a bioreactor during production of protein therapeutics. Now, when we grow these mammalian cells, they're grown in growth media that's rather complex, 50 or 100 different compounds. And in practice, if you could monitor what is in the growth media at any particular time, how much of these things are there, how much have been consumed, what other things might have been secreted, you can tell what's happening. You can monitor the progress of the bioreaction and understand things about it. So this is a metabolomics problem, and to do it non-destructively would be an excellent thing to apply NMR to, but superconducting NMR instruments are not very convenient to site with bioreactors. That said, though, we now have very good benchtop NMR instruments. And so it's within the realm of the possible to use benchtop NMR as a process monitor for a bioreactor. And this is work in progress by a postdoc here, Michael Judge in Subrata Mishra's group. Lots of you might know Michael because he trained with Professor Edison there at UGA. And so in the project, we're going to connect a bioreactor uh, in a flow system to a benchtop NMR instrument that will enable us to measure an NMR time series. And there are many different things that such a time series could be used for. Very simply, even if we don't analyze it in any particular way, a series of spectra that we measure during a bioreaction is like a signature of the bioreaction. And so we can use it just to compare the time series that we get with one batch to the time series that we measured with a previous batch to see if anything different is happening. And if we do more detailed analysis, we can do things like provide an early detection of failure or provide information that can be used to optimize the bioreaction with the ultimate goal of adaptive control. That is to be able to adjust the parameters of bioreaction in real time in response to an NMR measurement. This work is in its early stages, but what you can see here are some examples of 100 megahertz benchtop spectra measured over the course of about 2 weeks, 3 weeks. And without much trouble, we can easily identify a dozen different metabolites in this benchtop spectra. And I'm pretty sure we'll be able to almost double that number when we look a little more closely over time. Another interesting thing, which is a little bit unexpected but exciting, is at 100 megahertz on these benchtop instruments, we can also see a signal from the monoclonal antibody itself. Okay. Now, when you look at this signal on the benchtop, it does seem to be mostly a blob, but we've seen from previous studies that similar proton spectra that we measure, say, at 400 megahertz or even 600 megahertz have substantial information about monoclonal antibody structure. And we can use them to distinguish between different monoclonal antibodies. Just 1D NMR spectra at 400 and 600. And so when I look at these 100 megahertz spectra, they are blobs, but they do have features. So I'm pretty sure in time, we're going to be able to extract information from these as well. Now, these metabolomics-like problems made us interested in the same kinds of analysis that general purpose metabolomics folks are interested in. So we collaborated along with UA and Omid there and Professor Edison to make an analytical technique that could automatically identify and quantify peaks in a complex NMR spectrum. And that approach and software is known as SAND. I'm pretty sure all of you have heard about it before, so I'm not going to talk too much because we've had seminars over there at UGA about it. But a key idea is that we model the data in the time domain, and we use a cross-validation approach where we divide the points in the time domain data into a training set and validation set. And in our procedure to figure out how many peaks we need to model a particular region, we propose one peak and optimize it and test the results. Then we propose two peaks and optimize those and test the results, and so on, until, and each time we test the results by seeing how well those results fit the points in the validation set that were not included in the optimization. And when that stops improving, we know we've added enough signals. The method works pretty well. And one way that it was characterized by taking urine samples and spiking in different amounts of a 4-compound mixture. And using the mixture enabled us to very easily identify particular sets of peaks that we should follow. And we can see that as we add more and more of the spike in mixture, the intensities of the signals change in a linear way, which is just what we expect. So that's sand. Now, as a general point, we're interested to quantify NMR signals. And the way in which an NMR signal is generated is in the time domain. We measure a 1D signal basically with a radio antenna. And in a multidimensional experiment, we measure a series of these 1D spectra over and over again. Now, since we're measuring a 1D spectrum over and over again, the underlying signals are the same signals. They're at the same frequencies, but the details of the NMR pulse sequence are arranged so that the amplitudes of these peaks is modulated according to some interesting chemical information, like a chemical shift. So each one of these 1D measurements has the same peaks in it, but those peaks vary in intensity. They're modulated in intensity. And that holds true for the time domain, the interferogram, and the spectrum. So the mathematical implication of this is that a single signal in the time domain, even though it's a 2D matrix, can be represented as the product of a shape in one dimension times a 1D shape in the other. And for time domain data, it's a 1D time domain shape, which is a sinusoid, times a 1D time domain shape. So that gives us our two-dimensional time domain matrix. If we Fourier transform all the rows of this thing to generate an interferogram, the same property holds. The 2D matrix can be described as the product of a 1D shape times a 1D shape, but now one of the shapes is a spectrum and the other is still in the time domain. So now when we Fourier transform all the columns of this thing, we get a spectrum. But again, even though this is a 2D matrix, it can be described exactly as the product of these 2D, 1D line shapes. And the information content in all of these cases is exactly the same. In 3 dimensions, the same idea holds. So if we have 3-dimensional data that's still in the time domain in the 3rd dimension, that entire volume of a given signal can be described as the product of 3 1D shapes, depending on whether things are in the time domain or the frequency domain. If the first 2 dimensions are already transformed, we have a 1D line shape and a 1D line shape and a 1D sinusoid. Or if only 1 dimension is Fourier transformed, we have a 1D line shape and a 1D sinusoid and a 1D sinusoid. Exactly the same information in both of these cases. And NMR has other kinds of measurements that are independent collections of 2D measurements where the peaks stay in the same place, but the peaks evolve in some way. So for example, in a 2D relaxation series, we measure a set of 2D spectra where the intensities of the peaks is expected to decay exponentially according to dynamical properties of the molecule. Or we have a kind of experiment called a chemical exchange saturation transfer difference, or CEST, where we measure a series of 2D spectra in a way that helps reveal dynamical states that can't be observed directly. And the thing that both of these have in common is that they can be described by this signal model of the product of different 1D functions. Now, if we have a way to model the 1D shapes of a particular data set. Like let's say we can model a 1D line shape here and a 1D line shape here. That will enable us to describe the 2D spectrum. And for an interferogram, we can do the same kind of thing. We can model it as a 1D line shape times a 1D sinusoid and generate a model like this. The interesting thing about a model like this is that it doesn't have to be changed to accommodate non-uniform sampling. We can just forget about the parts that have been skipped during the measurement. And so what that means is if we have a modeling procedure that can be applied to interferograms, we can apply it to non-uniformly sampled data without the need to do any special reconstruction in the non-uniformly sampled dimensions. So let's see what kind of things that we can do by taking NMR data and decomposing it according to signals that we know are there already. And here's one example from the Edison lab. And Omid Sanity there helped design a carbon-13 detection probe with magnificent sensitivity, but also it gives a very curvy baseline. So in blue is this 1D spectrum that's generated by this fabulous curve. And actually, this spectrum is what started our collaboration many years ago. We can take a curvy spectrum like this one and do simple peak detection as a way to generate a very rough basis set of all the peaks that we can see here. And once we have a list of all of these frequencies, we can decompose this spectrum as a linear combination of all of these. But what we want to do instead is take advantage of the fact that this big baseline curvature comes from a distortion in only the first 4 or 5 points in the corresponding time domain data. So instead, we're going to take our basis set of peaks and inverse transform it to generate a time domain basis set. And then we'll go back and use this basis set to decompose the original time domain data and use the result to replace the first 5 points in the time domain to generate a nice flat spectrum like the one shown in yellow here. Here's another example. This is the first plane of a 2D relaxation series that has 10 planes. Each one of the peaks here represents one amino acid in the protein, and we expect these peaks to decrease in intensity over the course of an experiment. Now, we can use peak detection and line shape fitting to take this measured data and make a model like you see here. So this is a model generated by 2D Gaussian fitting. And the individual components of that 2D model are individual 2D Gaussian peaks like you see here. So that's what we're used to working with in the frequency domain. But instead of working in the frequency domain, we can take our experimental data in interferogram form and do the same with the model. So here's what the model looks like in interferogram form. And here's what the individual components in this 2D model look like in interferogram form. And we have a choice. We can either describe any plane in the data as the sum of these peaks, or we can describe any plane in the interferogram as a linear combination of all of these individual 2D interferograms. So this gives us a way to extract the intensities of these peaks on any of the planes in the data, which is the goal of the relaxation analysis. And in turn, what this allows us to do is extract data from a much more limited subset where more than where only an eighth of the original measured points are kept, and we can reproduce the intensities still within correlation factor of about almost 0.99 with 1/8 of the intensity. Whereas by conventional methods, we can usually go no fewer than 1/2 of the increments that we would usually measure. So we can decrease measurement time by a factor of 4. And now finally, here's a little project that's in progress. And just as a reminder to why it's important to us, when we have labeled material, we can conduct relaxation analysis on the monoclonal antibodies that we're so interested in. And one of the reasons that we would want to do that is to help understand formulations. So those are excipients, small molecules that are included with the therapeutic to improve its stability. And there are some kinds of excipients like salt that destabilize the proteins, and some like sucrose that tend to stabilize the protein. So these kinds of behaviors are known, but it's not known how they work, why they cause the effects that they do at the molecular level. And this is something that we can start to probe by NMR, by looking at changes in the overall tumbling rate of the monoclonal antibody or the internal flexibility. So we can do it with 2D relaxation series of the type that we analyzed previously. But to be able to get to a result like that, we have to assign all the peaks in these spectra. And here's an example of just the NIST Fab domain. Here's an excerpt of all the peaks from the light chain and all the peaks in the heavy chain. There's so many peaks that we can't label them all at once. So this is a Herculean effort to do this kind of peak analysis. And in the actual assignment procedure, for each one of these 2D peaks that you see here, there's probably 50 or 100 peaks in related 3D spectra that have to be analyzed and curated by an expert. So in a typical NMR workflow, a lot of time is spent by experts just manipulating peak tables and making decisions about what is a peak and what isn't, and whether or not two peaks line up with each other. And so for this reason, it's of great interest to us to find different ways to help automate the procedure of doing peak analysis. Conventional 2D peak pickers just look for local maxima, points in a spectrum that are higher than all of their neighbors, but procedures like this will miss partially resolved peaks, and they're likely to include small artifact peaks that really shouldn't. So we're interested to use machine learning to help us make determinations like this. There have been several labs that have tried this kind of work, and almost universally, when they analyze NMR spectra, they analyze the spectra as if they were grayscale images, which is a kind of logical thing to do because there's so much machine learning technology based around image analysis. So it's natural to try to exploit it, and to exploit it, you want to express the data in an image form. But when we actually look at an NMR spectrum as a human analyst, we do it on the basis of contour plots. And I would argue that to identify whether or not something is a peak, it's based on the shapes of these features, not their intensities. Okay, so being able to identify a big peak or a small peak, the absolute intensity doesn't matter. So instead of using these grayscale intensities, I'm going to suggest that we use contour graphics directly. And that's what we do. So we need ways to generate synthetic spectra. And there are a few ways that we can do that. One way is to start with measured data and identify a small number of well-isolated peaks, yank those out, make 100 different modifications by adding small amounts of line broadening or line sharpening or phase distortion. And then we have a big collection of physically realistic individual peaks that we can add together in different ways to make synthetic spectra that mimic experimental ones. And then we're going to use neural networks that are designed to perform segmentation. That is to say, to take an image and for each pixel, decide what that pixel is part of, whether it's in a dog, a cat, a stop sign, a car. This is such an important and widely used problem that there are many existing neural network schemes that have been designed and highly optimized with millions of pictures of dogs and cats. So we can take a network like this one that's already been trained on large numbers of images and just retrain a small part of it to do a particular kind of recognition. So we did that for our NMR data, giving contour plots as input, and originally using a scheme where we would ask the neural network to draw circles around the peaks that it would find. But it turned out that this particular approach is just as hard to analyze the result as regular peak detection. So over the course of time, we were able to generate a system where we could put in a contour plot and a collection of little dots where we know the true peaks are and get the neural network to reproduce the positions of those known peaks. So here's an example input. Here's the ground truth. Here's what the neural network tells us. And then finally, here's what a result looks like for a complete spectrum. So this is work in progress, but it's very promising and exciting. And it's a nice example of the kind of work that we do here at IBBR. So I hope that what we've shown you has been a convincing argument that there are lots of ways to use NMR to characterize biotherapeutics. And there are a lot of interesting projects going here at IBBR. Thanks very much. I hope this was of interest.
Thank you so much, Frank. So if someone has any questions, please unmute yourself or put your question on chat.
Hey Frank, that was a really great talk. So for that NMR technique that you described that you use to detect changes in higher order structure of monoclonal antibodies, is that sensitive enough to also detect uh, positional changes in unstructured regions of a protein?
That's a great question. And I would say it depends. I would say it depends on whether or not the changes in the structure result in some changes in the spectrum. So I could imagine
Gotcha.
structural rearrangements that happen on a time scale that they wouldn't be seen in the NMR spectrum. That said, if you can make some type of NMR measurement that does encode that information, then yeah, you could extract it with PCA and with related methods.
Thanks.
Frank, I have a follow-up question to that. So I really thought that the methionine oxidation of these antibodies was quite interesting and exciting. So my question was, how prevalent is, methionine oxidation in proteins in general, right? And the impact of this modification on protein dynamics. Is it unique to antibodies or is it appreciated as a more widespread
No,
mechanism?
it's an issue with every kind of protein therapeutic, and usually a specific therapeutic might have some methionines that are not at all related to places where the antibody binds to, and changes in those methionines won't have a clinical impact.
Hmm,
So generally, drug companies, while they're developing and testing antibodies, will identify particular residues that could be problematic for the clinical properties of the antibody.
I see. Thank you.
Yep. Yes.
Sorry
So
if you.
someone in the chat is asking about mass spec. So mass spec is used extensively for higher order structure characterization, and also mass spec is excellent at identifying chemical changes like oxidation. But the analysis to extract that information and the sample handling is very complicated because the protein has to be digested and separated. And even with the same exact sample, two labs will get different kinds of peptide fragments when they digest them and put them through columns. So unlike the NMR spectra, which are completely reproducible, mass spectra of these kinds of peptide maps differ from one measurement to another. So they're much harder to compare in a wholesale way. The advantage to NMR is there's no special sample preparation, and the measurements are conducted on the protein as formulated. So simple sample preparation, simple measurement, and no detailed analysis is needed to extract information for the kinds of cases that we looked at today.
So does anyone have any more questions? I don't see anything on the chat anymore.
Yeah, I'll just
Yeah.
ask if there's just a minute, Frank. That was great. Every one of your talks, I learn more and more and more. So this is why we keep on coming to you and
Thank
extending our
you.
collaboration. I'm excited to use. Is it Deco?
Yes.
The so the question, technical question, and it could just be a short answer. We can talk about it more, but you showed it working with absorptive peaks, line shapes. But what if you have like an antiphase peak, like for an inadequate spectrum or a cozy spectrum or something? Is there any limitation, or have you
No, there isn't. So um, NMR peaks can be modulated in amplitude, and sometimes they're modulated in complex amplitude, which means their height changes and their phase changes also. So, like you're saying, you get dispersive peaks. If the phase stays the same for each version of a peak in the interferogram, then you can use the method exactly as we have. If you expect the phase to change in every increment, you can use a version of this decomposition, which we haven't developed, that extracts complex coefficients for combining the basis set instead of real coefficients. And the complex coefficients would encode both the change in amplitude and the change in phase. Does it make sense?
I'll, yeah, and I'll send you a difficult data set sometime, and we can talk.
All right. And the person who developed DECO is on the line with us, Micah Smith. He's a computational biologist in my group. So you
It
could
looks
send
great, Mike.
condolences because he'll have to work on the complex decomposition too.
All right.
Thank
So
you.
thanks so much. Thanks so much, Frank, for joining us. And IOB folks, see you next week.
Thanks. Bye all.
Have a great weekend.
Talk to you soon. Thanks, Frank.
Bye.